Good day to Dr. Haryati. So today I will be presenting on the importance of fusion inhibitors and the ways they could be incorporated when designing drugs which can actually combat the influenza A virus, which has been a very upcoming conundrum across the globe, which really requires a very insistent and eminent sort of treatment to this, which the research community is currently working on. So before I present, I would like to give some key points for whatever that I'm going to present today, a light brief highlights. So first of all, we will look into the viral mechanism, the particular machinery, which actually how it really propagates throughout the cell and how it replicates and so on, and which machinery has been targeted by the scientist community on the whole by the fusion, by using infusion inhibitors, and how do they incorporate them into drugs, which sometimes have the potential to become successful drugs, and also the drugs which have been successfully established amidst the market. So first and foremost, as I said, the viral hemagglutinin machinery as an antiviral target. So this particular machinery has been targeted as an antiviral mechanism by the research community themselves to provide medicines, provide drugs to combat the influenza A virus on the whole. So let me brief. First of all, these viral hemagglutinin proteins, these particular proteins, they tend to have this particular domain that bind to the glycans that are found in the cell surface of the sialic acid, which literally happens over here. So when they bind, what happens is internalization of the virions via endocytosis will lead to the decrease of the pH, which means the entire environment tends to be acidic amidst the endosome. So this is the turning point, or this can be marked as the climax, which actually leads to the subsequent effect or the tailing effect, the repercussions, which is caused by the virions, which actually tends to lead to the final replication, because this particular acidic environment could lead to the irreversible conformational change of the HA protein. So when there is a conformational change, the fusion protein, the fusion peptide of this particular HA protein is exposed. When it is exposed, this triggers the fusion with the endosomal membrane of host cell, and thus the viral ribonucleoproteins are released into the cytosol. They migrate to the nucleus for subsequent replication, and the entire cell is done by the virus itself. So scientists have targeted two important factors here. One is blocking of the HA binding to the sialic acid residue, which is the beginning or the initiation of the entire process itself. Or another target could be the blocking of the HA conformation change, which roots out from the acidic environment of the endosome itself. Hence, this is particularly the rough idea of what really happens due to the HA mechanism of the virus itself. So the internalization of virion could increase the acidity in the endosome, which will lead to the conformational change, and thus the repercussions would be the membrane fusion followed by replications. So what do scientists do literally, and what do they have discovered is that this particular fusion inhibitors, how do they function? Now, they function by preventing this acid-triggered conformational change, which then enables them to incorporate these fusion inhibitors when designing drugs as the antiviral drugs to combat influenza A virus. So, the first fusion inhibitor, which I'd like to talk about, is B7 cyclic peptide, which is currently on the stage or the status of being a potential fusion inhibitor to be incorporated into drugs to combat influenza A virus. So from a very pivotal anecdote, P7 cyclic peptide is well prominent, well eminent for its specificity. Why? Because the structure of this peptide is solely conjectured on the note of the heavy chain complementarity determining region loop of BNEB F16V3 and framework region 3 of CR9114. So these components in this particular cyclic peptide makes it more specific, which makes it to be highly eminent and a highly potential candidate to be incorporated for drugs that are actually against influenza A virus. So, looking in depth, note on a very in-depth sort of deliberation. P7 literally binds to the HA stem epitope. So this is where it hits the key point, the climax, where it blocks the low pH, the acidic pH, which is actually the prime factor which causes the conformational arrangements, followed by the membrane fusion, followed by replications. So this is particularly the first step in which the P7 cyclic peptide attacks or combats. It combats the low pH induced conformational rearrangements first. It blocks that entire process. by its specificity, by binding to the highly conserved HA syn epitope. So what makes it P7, what makes P7 super specific for group 1 HAs? It's because of the glycosylation of ASN38 in group 2 HAs and substitution of tyrosine 49 group 1 for a larger ASN group 2 that improves binding. These groups are literally which belongs to the HA itself, two different types of HAs, okay? So this particular figure here, it shows the structural elucidation of the group 1 HA specificity of the peptides. Moving forward, Arbidol. Now, Arbidol is one of the most successful fusion inhibitor, which has been incorporated into drugs successfully amidst the market to prevent influenza virus, to combat influenza virus, and it's one of the most successful feats by the scientist community themselves. So, applauses to them. Well, when we come to Arbidol, this particular topic, literally we will be focusing more on the hydrophobic cavity found in the HA trimer. This hydrophobic cavity in the HA trimer would be literally targeted by this particular Arbidol. So Arbidol works by actually targeting the hydrophobic cavity in the HA trimer, which is the particular place where it binds. So this particular binding literally increases its specificity on greater levels, on greater heights. So where does it bind? Let's look into a closer note. Arbidol consists of peptides that function as fusion inhibitors such as F1, F5, F7, and F8. So all these components all these peptides, they tend to bind in the hydrophobic cavity in the HA trimer stem at the interface between two protomers. And binding of this Arbidol to the cavity, hydrophobic cavity in the HA trimer will literally it will make some hydrophobic interactions for sure, and it will induce some conformational rearrangements to form a network of inter and intraprotomal salt bridges, which will then subsequently or concurrently, it will literally block the pH-induced conformational rearrangements. So it will block the entire network by causing a different sort of conformational rearrangement, which is the formation of this particular intraprotomal salt bridges. So by that, it stabilizes the perfusion conformation of HK that inhibits the large conformational rearrangements, which will lead to the membrane fusion. That's the functions of, those are the functions of Arbidol. So Arbidol has a very high shape complementarity with the binding sites in these two groups of HAs, group 1, group 2. It has a very high complementarity due to the presence of F7 and F8 that work as a minor fusion inhibitors. So all these structural informations, all these opulent informations on Arbidol has really enhanced or enabled the scientist community to come up with superfluous drugs that can combat influenza A virus on a very high note. So incorporation of these sort of fusion inhibitors are really imminent and really significant, and the scientist community is still conforming with its efforts going forward and progressing with its efforts by literally researching more in depth on the structures of Arbidol, P7 cyclic peptide, and many sorts of other fusion inhibitors to be incorporated into the drugs and to come out with really efficacious drugs to put an entire full stop to the influenza A virus, though it might sound a little, well, perhaps not necessarily impossible, but let's just hope for the best and let's save our globe from this particular influenza A virus. Thank you.